DNA polymerases make copies of DNA molecules by adding dNTPs to recessed 3' DNA ends. There are multiple DNA polymerases operating in both eukaryotic and prokaryotic cells that play different roles within the DNA life cycle. In E. coli, DNA polymerase I is involved in DNA replication and DNA repair. It's encoded by a single polypeptide. The protein has multiple domains responsible for different aspects of its overall reaction, including a distinct domain for proofreading. This domain is removed in clenal fragment of DNA polymerase I. Most thermostable polymerases on the market today are homologs of full-length DNA polymerase I or of its clenal fragment. Like with the exonucleases that degrade a DNA, the different enzymes that polymerize DNA have diverse and specific subtle variations in behavior. I'll go through what the different properties here mean. Many of these variant behaviors of polymerases are about how they respond to nicks. Suppose we have this DNA involving 3 polynucleotides annealed together such that there remains a single 3' recessed end. Any DNA polymerase will extend this recessed end. But what happens when it collides with another annealed DNA? If the polymerase has 5' to 3' exo activity, it will chew it up. The DNA strand is ultimately entirely replaced by new synthesis. The more common scenario is strand displacement. The polymerase synthesizes DNA until it reaches the blocking strand, and then it displaces the blocking strand. That ultimately results in a DNA that is released as a separate complex. The enzyme Phi29 is the most robust strand displacing enzyme, and it's one of the more useful molecular biology tricks to do rolling circle amplification with Phi29. First, the DNA is denatured to separate the strands. I'll just focus on one of the strands here. This allows the annealing of oligonucleotides that can then initiate polymerization. The new strand eventually reaches its own 5' end and will undergo strand displacement. The newly released strand can initiate further polymerization, allowing rapid isothermal amplification of the original DNA template. Another example of the utility of subtly different polymerase properties is Kunkel Mutagenesis. Kunkel involves the use of T4 DNA polymerase, which has neither 5' to 3' exo nor displacement behavior. An oligo that is homologous to the DNA but contains a mutation designated by the red X is annealed to the DNA. It then undergoes polymerization around the circle. The DNA is then transformed into E. coli, which can repair the nick. If a different polymerase were used that did strand displacement or exo, the X would become dislodged or degraded and would not be incorporated into the final sequence. Polymerases can also have 3' to 5' exonuclease activity. This is the property referred to by proofreading in the context of polymerases. It is the major component of the enzyme that determines its fidelity. It is absent in Taq-like enzymes, which also have higher error rates. It is present in PFU-like enzymes. Enzymes with this activity will produce blunt-ended PCR products, while those without this activity will typically contain a single 3' A overhang. This document, the Thermostable Polymerases, goes into further detail about the subtle differences between the commercially available thermostable polymerases organized around specific use cases. Reverse transcriptases are enzymes from retroviruses that synthesize DNAs from an RNA template. A DNA oligo is annealed to the RNA, and then the enzyme will polymerize it to its end. Finally, there is terminal transferase. It is a template-independent polymerase that operates on protruding 3' or recessed ends, blunt ends, and on double-stranded or single-stranded DNAs. It has no exonuclease activity. It is used primarily for radioactive end labeling or for adding homopolymeric ends to a DNA, such as a poly A sequence.